My name is Sarah Anderson, and I am a PhD candidate in the chemistry department here at Northwestern. And today I'd like to talk to you about the fault in our tools and a new analytical tool that I've developed to accelerate the process of drug discovery for brain cancer. Last summer, you probably heard the word glioblastoma in several news reports upon the decline and death of American politician John McCain. Mr. McCain was diagnosed with the aggressive class of brain cancer in July of 2017, stopped treatment on August 24th, 2018, and passed away the very next day. While this timeline might seem exceptional, the unfortunate reality is that Mr. McCain's experience with glioblastoma was very representative of the rapid progression of the disease. In fact, those with glioblastoma survive, on average, just over 1 year upon diagnosis. With the goal of developing treatments for glioblastoma, scientists examined glioblastoma cells for any irregularities that could be targeted by drugs. They discovered that glioblastoma cells contain more of the enzyme isocitrate dehydrogenase 1, or IDH1, relative to normal cells. Enzymes facilitate specific chemical conversions within the cell, and IDH1 helps to convert a molecule called isocitrate into a different molecule called alpha-ketoglutarate. At the same time, it converts a molecule called NADP into another molecule called NADPH. The cell uses NADPH to produce precursors that build the cell membrane. And scientists believe that by having more IDH1, glioblastoma cells overproduce NADPH and are therefore able to divide more rapidly than healthy cells, forming cancerous tumors. Now that we understand the role of IDH1 in contributing to the progression of glioblastoma, is there anything we can do about it? The most effective way to counteract the effects of excess IDH1 in glioblastoma cells is to use an inhibitor, which is a small molecule that binds to the enzyme and impairs its ability to convert isocitrate to alpha-ketoglutarate and NADP to NADPH. In order to evaluate if a molecule is a good inhibitor of IDH1, a scientist can measure how much alpha-ketoglutarate the enzyme produces in its presence. The less alpha-ketoglutarate produced, the better the inhibitor. The tool that is currently used to measure alpha-ketoglutarate in the cell acts like a funnel, in that all of the molecules present in the cell go in, but only alpha-ketoglutarate comes out. This strategy requires separating the many different components of the cell by their chemical properties in order to isolate the alpha-ketoglutarate molecules for detection. As you can imagine, this process is very arduous, and only one sample can enter the funnel at a time. As a result, this tool can analyze a maximum of only 18 samples per hour. There are thousands or even millions of molecules that could inhibit IDH1, and testing them all using this tool is delaying the discovery of an effective IDH1 inhibitor. My research focuses on minimizing this delay by developing a new tool that can measure alpha-ketoglutarate from the cell as an indicator of IDH1 inhibition, but that is also capable of rapidly analyzing a large number of potential IDH1 inhibitors. The tool uses a plate that is coated uniformly with molecules, forming what is known as a monolayer. The molecules in the monolayer are designed to undergo a chemical reaction with alpha-ketoglutarate that captures it to the surface. The plate is then analyzed by mass spectrometry, which is an experimental technique that reads the mass, or weight, of each molecule in the monolayer and produces a peak at its mass value in the resulting mass spectrum. The intensity or height of the peak corresponding to the monolayer alpha-ketoglutarate reaction product is proportional to the amount of alpha-ketoglutarate captured to the surface. This tool is faster than the technique traditionally used to measure alpha-ketoglutarate in the cell for two reasons. The first is that the monolayer's chemical reaction occurs almost exclusively with alpha-ketoglutarate, like a piece of bait that's used to attract a certain type of fish. By quickly isolating alpha-ketoglutarate from the other molecules in the cell, we save time on extensive separation procedures. The second is that this tool can accommodate hundreds of samples at once. The surface of the plate is arranged in a 384-spot format that is compatible with automated liquid handling instrumentation. This means that up to 384 samples can be prepared and transferred to the plate in tandem. Analyzing the plate by mass spectrometry, which takes about 3 seconds per spot, provides mass information that can be used to calculate the amount of alpha-ketoglutarate present in the sample applied to each spot. This tool therefore enables a comparison of the efficacy of over 1,000 IDH1 inhibitors per hour, making it ideal for the rapid screening of large libraries of glioblastoma treatment candidates. Preliminary experiments using this tool have produced promising results. In order to evaluate its ability to accurately measure alpha-ketoglutarate in the cell, I used it to test cells that had been engineered to produce less alpha-ketoglutarate. The tool measured a significant decrease in the concentration or amount of alpha-ketoglutarate in the engineered alpha-ketoglutarate deficient cells relative to the normal cells, and the degree of decrease observed was consistent with results from more established experimental methods. While still in its proof of concept stage, this result suggests that this tool can one day be used to assess the potency of IDH1 inhibitors for glioblastoma therapy. Because of the dependence on slow analytical tools, long drug development periods are nothing new. In fact, it takes on average 12 years from a drug's conception to its commercialization. But the average time from glioblastoma diagnosis to death can fit within that 12-year period almost 10 times. If there were ever a disease that could benefit from accelerated drug discovery, it's glioblastoma. By developing a tool that can selectively capture alpha-ketoglutarate from many samples simultaneously and quantify alpha-ketoglutarate concentration using mass spectrometry, I hope to reduce the amount of time needed to discover an effective IDH1 inhibitor and a potential treatment for glioblastoma. Thank you.